James, you shot your pistol into the air and I'm off. All right, well, howdy everybody. I've got a topic that I think should be fun to talk about, but also incorporate a lot of things about diversity and phylogeny because that's sort of my bread and butter. I am a beetle taxonomist. I specialize in the family Lyotidae. These guys are also called round fungus beetles or truffle beetles. They are lovely beetles. As you can see from this assortment, they come in a whole rainbow of different colors of brown and a variety of interesting body shapes. There's about 3,000 species of them worldwide. They're just a lovely, bizarre little beetle family. And they do have a bit of a reputation amongst other beetle researchers for being sort of these little brown beetles that are about the size of a pinhead. We call them LBBs, and they're sort of often considered one of the more taxonomically challenging families because they're mostly about 2 millimeters long and are all about the same size, shape, and color. So this fascination with Lyotids started back when I was an undergraduate doing honors research, and I was collaborating with a professor who also did beetle taxonomy, and we were describing some new species in the genus Egliptinus. And we were describing them because they were obviously an exciting new species. They had these fabulously bizarre antennae that you can see on the screen there. And while I was doing this research as an undergraduate, we also noticed that some of these species were not really as brown as little brown beetles are supposed to be. As a matter of fact, they were iridescent. And this is rather exciting when you're used to looking at things that are 50 different shades of brown and suddenly you find something iridescent. You say, hold on a second, what's going on here? How are they making that iridescent? What's happening? I've got to get to the bottom of this. And so I was just doing my taxonomy, just describing new species, the kind of science that a lot of people sort of looked at on as stamp collecting, and came across this really unusual optical phenomenon. And when you take these beetles and you put them under a scanning electron microscope, you'll notice that the ones that are iridescent have these little sort of specks on the surface of the elytra. And if you zoom over and take a picture of that with the SEM and dorsal view, you see all these little parallel tiny ridges. So this was clearly the structure that was leading to the iridescence in these tiny brown beetles. But I wanted to figure out exactly how. And that took me onto a long journey of studying beetle iridescence, which I'm about to drag you along on with me. Iridescence in nature just refers to, if iridescence researchers can agree on anything, any color that appears to change in hue with a shift in the angle of illumination or observation. So more for butterflies, peacock feathers, golden scarabs, as you move your head back and forth, their color will seem to change. That's what we call iridescence. And if you're an iridescent animal, you'll never be making that color using pigment. It's a type of structural color, which means that the color itself that we perceive arises from the way that photons interact with the nanoscale structure of the cuticle, so of the nanoscale structure of the feathers, of the wing scales, of the elytron of the beetle. And when you're a beetle, there are essentially 3 different ways to be iridescent. The little Lyotids that I was studying are doing the very first way, the first way that I'd like to describe, which is something we call diffraction gratings. A diffraction grating, like we see in the Lyotid, is any series of parallel slits or ridges that can disperse white light into its constituent wavelengths. So the white light hits this grating and it gets scattered out as sort of a rainbow. We call this spectral iridescence, like a spectrum. If any of you are old enough to have ever seen a CD-ROM, when you look at the underside of it, it has that sort of a rainbowy color on the underside. That is because the little grooves that have recorded the data in the disk also act as a diffraction grating. And in beetles, there turns out to be a really high diversity of different types of diffraction gratings. This is from a little bit of side research I did as a graduate student. You have very simple gratings that are simply just a parallel series of slits that disperse the white light that you shine on the beetle into a sort of a smear of rainbow color. So this is a very simple example of spectral iridescence. You see what are called blazed gratings that have a sawtooth profile. You see these very short parallel ridges. This is the kind of grating that we documented in those Lyotids. You see what we call a waffle grating, which is essentially a bidirectional diffraction grating. I found this in sap beetles in the genus Palodes. And then some of the very weirdest diffraction gratings are sort of these fringes or tabs you'll see in sericine scarabs. So these are extremely common, extremely tedious looking brown pasture chafers. And when you get the right ones under a microscope and shine some light on them, you'll see this beautiful rainbow iridescence. And that's coming from a completely different sort of diffraction grating. And so it turns out these diffraction gratings are actually really, really common in beetles. And we're not entirely sure what they're for. On the other hand, one of the most common types of beetle iridescence you'll see, and when you think of an iridescent beetle, you'll think of this, is what we call a multi-layer reflector. This is simply a stack of layers of chitin, where the thickness of each layer of chitin in the exoskeleton is about 1/4 of a particular wavelength of light. And when you hit that stack with white light, it will reflect that one particular wavelength with what we call constructive interference. That means you get very strong, brightly colored reflections. So for example, if you look at this dog-bane flea beetle that's in the illustration here, it's got a bright blue pronotum. That's because the layers of chitin in its exoskeleton on the pronotum happen to be about 1/4 of the wavelength of blue color. And so what you get is this really bright, vivid color that will essentially last forever. There have been fossil beetles dug up that are still iridescent because this structure has been preserved even despite the fossilization. And I will move along. Here's a nice little TEM image of a cross section of a multi-layer reflector in a beetle. This is the tortoise beetle, Aspidomorpha tecta. And it's a lovely sort of golden color, and that is arising from this multi-layer reflector, which you can see those sort of stacked layers in the exoskeleton. Now, those are the two probably most commonly encountered types of iridescence in beetles, and they're pretty neat. There are a lot of questions we can ask about diffraction gratings, but the third type of beetle iridescence is the one that was most exciting to me because this is something that was discovered relatively recently and represents a technology that humans only got around to developing about 20 or 30 years ago. So that is what we would call a three-dimensional photonic crystal. It's also called an opal analog because it is structurally analogous to opal. So on the left, you see a scanning electron microscope image of the, I believe, silicon dioxide nanospheres in an opal, the mineral, the gemstone. And on the right, you see the first SEM image ever taken of the opal structure that gives these beetles their outrageously bright colors. We sometimes call these guys the party weevils or the disco weevils because they are so glittery and extravagantly colored. So after that was described in 2003, more papers started popping up where some people with an engineering background would get their hands on one of these so-called opal weevils, and they would slice open these tiny little scales, these little sort of round lenticular scales that are growing from the back of the weevil. And when you cut them in half, you see this beautiful lattice framework. This is the photonic crystal. And the photonic crystals produce these incredibly bright, dramatic, reflective structures. But when you have enough of them with crystal domains that are oriented in different directions, you'll end up with an overall structural color that's not iridescent. If you look at Entamus imperialis on the bottom image there, that is a beetle whose little scales are full of these incredible photonic crystals, creating incredibly bright reflectance. But because there are so many different small domains of the crystal, the overall effect is to give you a bug that's green. So this is a way to be cryptic if you're sitting out there eating leaves without having to be iridescent and without having to make your own pigment colors. So this is really exciting. Humans didn't figure out how to fabricate photonic crystals until the late 1970s at the very, very earliest. So a quick aside about the dimensionality of the photonic crystals. A one-dimensional photonic crystal simply has periodicity in one dimension, along one axis. So the multi-layer reflectors that we discussed would be a one-dimensional photonic crystal. A two-dimensional photonic crystal would be sort of what we'd call a woodpile structure. And then the three-dimensional photonic crystal that we're discussing in these weevils has periodicity on 3 different axes or 3 different dimensions. And this is also really cool because three-dimensional photonic crystals can be used for all kinds of things, especially if you're making them out of something like chitin, which is just a relatively simple polysaccharide that's very lightweight and biocompatible. And there are already quite a number of groups working on bio-inspired or biomimetic engineering based on the photonic crystals that we have seen in beetles. So what I wanted to know as a person who's a beetle person and not an engineer is, how are the beetles making them? How do they make them in a developmental sense? And how did they make them in an evolutionary sense? So I and a couple of collaborators from Yale University set out to ask, how are beetles making photonic crystals? So we want to look at both the phylogeny and diversity of the beetles and the diversity of their crystal structures to try to find the answer. So the first thing that we did is try to characterize the geometry of these three-dimensional photonic crystals inside the scales of the weevils. The reason we want to do this is because if we have an idea of the different types of crystal geometry that they can make, that'll help tell us what kinds of chemical or material constraints are acting on the way that they develop. So the first thing we did is we went down to the Australian synchrotron and blasted a beam of extremely high energy X-rays directly through the crystals of the weevils. This was one of my favorite projects, where I got to take a series of weevils from the Australian National Insect Collection and spend many late nights in the lab painstakingly shaving off their tiny little hairs and enclosing them in little sandwiches of mylar film so that we could shoot the X-rays through them without too much interference. Here's the Australian synchrotron, which is really lovely. It's a little particle accelerator. And there's little baby me about 15 years ago, I suppose, with an extremely large array of computer monitors adjusting the aim of the X-ray beam that we're blasting these little weevil scales with. And what happens is you blast the weevil scale with the X-ray beam and you will get a diffraction pattern, which you can see there at the left. And this diffraction pattern tells you exactly what geometry of crystal you are dealing with. And so we did this for a selection of weevils across the whole diversity of weevils that have any kind of scales with an interior that could be crystalline. And the almost unreadable published result was a lovely sort of survey of the diversity of biophotonic nanostructures, or weevil photonic crystals. What we found is that there is a tremendous diversity of photonic crystals, of three-dimensional photonic crystals in insects, and especially in beetles, more than we had originally expected. And you can see they form all of these different sort of shapes. And as a matter of fact, you see quite a lot of different crystalline geometries. Each of these different names corresponds to the way that the nanospheres, either air nanospheres or chitin nanospheres, are arranged inside the scale lumen of the weevil. And you'll notice that there's diamond, there's gyroid, there's simple cubic, face-centered cubic. These are all different ways, if you imagine stacking up a palette of oranges. These are simply different ways to arrange those spheres in space in a repeating structure, in other words, in a crystal. And the chemist I was working with took a look at this and said, oh, you know what's interesting about all these different crystalline geometries that we've observed is that all of those are possible lyotropic phases. And if you're like me and you said, excuse me, what's a lyotropic phase? I want you to imagine making a bottle of vinaigrette salad dressing. You've got your olive oil and you've got some vinegar, and you're going to put them in together and they'll be separated. Just shake it up really good and you put it on your salad, and then you set the bottle down and they will separate back out. That is a lyotropic process. Whenever you have an amphiphilic molecule in a polar solvent, kind of like the little phospholipid molecules that make up our own cell membranes, they will self-assemble into a variety of different structures, which I've artistically represented here as these blue shapes. All of those are shapes that any amphiphilic molecule will self-assemble into depending on its concentration in the solvent. So what that suggests to us is that there's a very good chance that these photonic crystals in the beetles are not formed through some kind of biotemplating. They are forming through some kind of lyotropic process, which to my knowledge is the first time that that's been documented in a structure that's made out of chitin. So we have a clue about how they may be making these incredible photonic crystals. So it appears to be something like a lyotropic mechanism, and it only shows up, these photonic crystals only show up within insects, especially beetles, that have scales. So all insects have little hairs or setae. You'll see them on flies, you'll see them on grasshoppers, on odonates, on everything, but only in beetles where those hairs have been evolutionarily modified into a little flattened seta with a hollow space in the middle. Only there do you see the photonic crystals. So this suggests that that's something about that self-contained environment allows you to have that sort of that fluid matrix in which this kind of self-assembly can happen. So this is a case where we haven't yet been able to rear these weevils through larvae, through pupation, and arrest the pupation to actually catch them in the act of lyotropic self-assembly, but we can infer that it may have happened by looking at the actual geometry of their crystals. So that's something that's tremendously exciting to me and some of my beetle friends, and hopefully maybe a few of you have had enough chemistry classes to also be excited about that. Now we have an idea, as I've just explained, of how the beetle photonic crystals assemble during the development of the insect. So essentially during pupation, we have that self-assembly apparently taking place inside the hollow interior of the scale on the insect. The other thing that we wanted to do is ask, how did these crystals originate from an evolutionary perspective? We seem to have at least two origins of three-dimensional photonic crystals in phytophagous beetles. One in the longhorn beetles, or cerambicids. Those are the guys that you can see up at the top with the very snazzy pinstripes. They have a couple of different geometries that we see in their photonic crystals, but then also in the weevils, the curculianoids. And so because we happen to have the data for the phylogeny of weevils that a friend of mine at CSIRO was working on, we thought, okay, let's construct the phylogeny of weevils, map what we know about the crystal geometry on there, and see if we can reconstruct its evolutionary history. So that turned into a paper that just came out in 2019, and I will walk you through how we figured that out. So our question in this paper was, how did we go from little brown, boring weevils to these incredibly flamboyant party weevils that you see on the right there? That's an example of Pachyrhynchus, who are using three-dimensional photonic crystals to make these incredible colors. What we did is first we reconstructed the phylogenetic history of weevils using some new data, as well as a little bit of previously published data for about 64 species sampled from all of the major lineages of weevils. So if you look across the whole family tree of weevils, all of them will have some setae. Only some of them have flattened hollow setae that we would call scales that would have they'll either be clear, brown, white, or colorful. And when we reconstructed the history of those characters, starting by distributing them across the tree and then performing an ancestral state reconstruction, we found that three-dimensional photonic crystals appear to be what we would call a synapomorphy, or a shared ancestral character not present in any other group, for a clade of weevils called the Entomini sensu-latu, which just means the broad-nosed weevils and their friends. There are squillions of species of weevils. I'm not going to try to give you an exact number, but it would be upwards of 200,000 at this point. And they don't all have the photonic crystals. There's just this one particular clade. There's this one lineage with a shared common ancestor in which we see these three-dimensional photonic crystals arising. And if you go through the entire weevil phylogeny and you code, sort of make yourself a cladistic matrix of morphological characters looking at the the types of CD that that weevils have, you'll see that there seems to be an actual progression. You see sort of an ancestral at the base of the weevil tree. You see this ancestral transition from slender CD, regular insect CD that are hair-like, to a sort of a rounded scale. And then the interior of that scale starts out as a spongy matrix that just scatters light essentially randomly, which gives you a whitish color. And then at the base of the broad-nosed weevils, that transitions to this crystalline lattice that's capable of producing a very high intensity reflectance of a particular color, such as green or blue or what have you. And then within the weevils, within those broad-nosed weevils, we also seem to see at least one transition from a diamond geometry crystal to a gyroid geometry crystal, which is a slightly more complicated type of photonic crystal. And so there seems to be this very clear sort of progression of steps that you have to take if you are a lineage of weevils and you want to evolve a photonic crystal. And when you pair that with what we know about the ecology of weevils, about the evolution of weevil feeding ecology, there are some very strong correlates that suggest what the benefit of these photonic crystals might actually be. So at the very base of the weevils, you have things like anthribids, also known as fungus weevils. They are crawling around on brown fungusy logs, eating white fungal growths and hyphae. And for them, you would expect to see simple scales with or without a white scattering matrix inside the scale lumen. This would give you the benefit of being cryptic in a fungusy wood habitat. And this correlates extremely well with what we know about the evolution of weevil feeding ecology. Then the base of the entomine weevils, where we see the origin of those three-dimensional photonic crystals, also happens to be a major inflection point where that lineage of weevils shifts from feeding on fungus to feeding on plants. And so you see this origin of very strong association with angiosperms and crawling around on green land plants. And that happens to be about the same point at which they evolve these photonic crystals that reflect predominantly in the green. And then finally, the question we're still asking is, among the extremely colorful weevils that have gyroid type crystals, what are those for? Are they aposematic? There's some indication that they might actually be warning predators that, look out, I'm a weevil with extremely strong, thoroughly fused elytra. I have super tough body armor. You can't eat me. And this is there's a whole clade of weevils called the pachyrincine weevils in New Guinea and the Philippines that actually appear to be doing this. It could be mimicry as well. It could be mate recognition. We haven't been able to suss out why they're doing it yet, but it seems to be much more of a conspicuous visual signal rather than an attempt to be cryptic in an angiosperm ecological context. So to sum up, I want to talk about what we know now about beetle three-dimensional photonic crystals that we didn't going in. So we see an unexpectedly high level of structural diversity of different crystalline geometries, all based on the same toolkit, which is a chitin or polysaccharide-based hollow seta that assembles a crystal during development. It has to be a hollow seta with a contained setal lumen in order to develop. We never see photonic crystals that are not in that exact context. They don't form them inside the exoskeleton, inside the smooth part of the elytron. They always form the photonic crystals inside the seta. So because of that, combined with the exact suite of different types of crystalline geometry we see, we can be reasonably sure that these crystals are being formed during pupation as a lyotropic phase separation process. In other words, again, you have those amphiphilic molecules that are self-assembling into these different crystalline shapes and geometries in a fluid during development, during pupation. And then the way that this appears to have evolved is through this transition in the weevils from fungus feeding on the ground in concealed environments on fungusy logs to leaf feeding, angiosperm feeding out there on bright green vegetation and needing to be cryptic. So that's how it seems they develop in pupation and how it seems that they have evolved on a larger scale. And with that, that brings me to the end of my talk. There are a lot of extra questions that we still have to ask about these guys that we're really excited to figure out. But that is my story of how I went from being a little brown beetle taxonomist to sussing out exactly how photonic crystals have evolved and what they seem to be used for in these outrageously flamboyant weevils. So thank you so much.
Thank you, Ainsley. Thank you very much. That's absolutely awesome. What a cool study system. Yeah, I'm brimming with questions. Does anybody we haven't had any questions posted in the chat yet, but if anybody's got any questions for Ainsley, please do feel free to type them in or to unmute your mic and go for it. I'll ask one to start with. I just love the idea of the crystals self-assembling like a vinaigrette. That's brilliant. And so the idea is they do it while the beetle is undergoing metamorphosis. Exactly. In the pupa. And they do it within the CT of the hairs, right?
Mm-hmm. That's correct.
Hairs. Yeah, could you potentially like experimentally screw with that and get like a custom colored beetle?
Yes, you could. You would have to identify what is governing the density of these, whatever these amphiphilic molecules are inside the CD. We assume that they are a chitin molecule with some other type of molecule affixed to it, or some tail or something. An amphiphilic molecule has one end that's hydrophobic and one end that's hydrophilic, right? And so the hydrophobic end wants to get away from water, from a moisture that it's suspended in, and the hydrophilic end wants to be with it, with the water. And so if there is a fluid environment inside these scales as they develop, which we know there is, then you'd want to kind of be able to create more of those amphiphilic molecules during pupation. And so the trick is, the reason that I haven't been able to try to do this before, is that these weevils, these fascinating, wonderful weevils, the entomines, the broad-nosed weevils that have these photonic crystals, every single one of them undergoes pupation underground, usually attached to the roots of a tree. Okay. And the only way, at least in Canberra, when I was based down there, the only way to get one was to wait around until one of your neighbors was digging up an entire wattle tree from their front yard and then dive in and get the pupa. That was the source of all of the pupae in our collection. And that's not something that happens. So it's a bit challenging. It's hard to replicate that in the lab. Now, there are some entomine weevils that develop on the roots of clover, and that's a lot more realistic of a system to work with. The other problem is that when you're looking at insects that undergo metamorphosis, they essentially turn into soup inside the pupa and rearrange their whole bodies. They'll have little, what we call imaginal discs, which are just little clusters of cells that are going to become adult structures like antennae and mouthparts and legs and genitalia and wings. Those are always in the larva waiting, but those are just tiny clusters of cells. And when the larva pupates, it forms its tough outer pupal shell, and then the interior of it, everything except the imaginal discs, turns into soup. And so if you take a pupa that's too fresh and you cut it open, out comes bug soup. It's really difficult to do something like a transcriptomic analysis and find out what genes are being expressed in a very particular tissue when there's not a particular tissue. If you do a pupa that is too late in development, you will pull out the elytron and the crystals will already be formed. And so you have to pinpoint the exact point in development to find out what genes are getting expressed inside those little CD as they develop. And then those are the genes that you can mess with in order to fabricate your own crazy colored weevil. Wow. The easier way to do it is just get your own reagents. Go to a polymer chemist and say, tell me, I want to self-assemble some photonic crystals. I want to use a polysaccharide like cellulose or keratin or chitin, and they call it a block copolymer. And we're going to just have that self-assemble by mixing these, our amphiphilic molecule of our choice and the polar solvent of our choice. And they're already doing that. So we first tried what's called biotemplating, where you simply take something and squish it against a beetle photonic crystal at the very, very small scale. And you've made a template, and then you can make a cast from that of a photonic crystal. That's been successfully done. But they are also now basically fabricating their own photonic crystals out of polymers based on this particular these structures that we see in weevils. It's much easier to do it in the lab, thoroughly outside of the insect. So
okay,
that's where we are for now.
Cool. So we've got a couple of student questions here. I don't know if you can see the chat.
Let me see.
Fiona asks, what are the? Do you know what any of the possible applications for this for the CNC biomimetics and
oh, absolutely. There's so many wonderful applications. First of all, when you're trying to make a display or a picture or a LCD screen like you might have on a phone, when you want to make a display, right now we're making those out of glass and chemicals and so forth. And wouldn't it be nice if we could make a lightweight, flexible, non-toxic, biocompatible display out of polymers like chitin? We're already starting to get quite good at making bioplastics by taking animal chitin from shrimp or from insect exoskeletons, basically melting that down and rearranging the polymers in the shape that we want. There have been wound dressings made out of insect-based chitin that way. There's people making biodegradable plastics that way. And if we can do that kind of fabrication with chitin, which is relatively unsophisticated, if we could find a way to fabricate photonic crystals, three-dimensional photonic crystals out of chitin, we could be making, almost essentially disposable, super lightweight, biodegradable displays, colors, warning signals, that kind of thing.
Oh, that's amazing.
So that would be really exciting. Go ahead.
How cool. Okay, right. So let me get these in order. So Jennifer asks, and actually this is related to a question that I was going to ask. So is there any evidence for sexual selection acting on the iridescence? Seeing as the colour and patterning is fully developed by the time that the individuals are mature enough to be searching for mates. And if I might, I'm going to append a question to that, which is there any evidence that the iridescent colours need any kind of maintenance by the beetles? Like, are they costly in any way to make?
They are as cheap as can be. It's much more costly to make pigment colors, because in pigment colors, you essentially are turning your body into a little biosynthesis factory where you get these chemicals from the plants that you eat, and you them and express them in your exoskeleton. And it seems to be more metabolically expensive to make a pigment color than to make a structural color. They do not require any maintenance whatsoever. Sometimes people will look at beetle specimens in a collection and say, oh, do you polish this? Have you put varnish on this? No, it's just that beetles are essentially a sort of a RoboCop type of animal where their exoskeleton is formed of transparent cross-linked chitin microfibrils that are almost exactly like Kevlar, in terms of structure, strength, and flexibility. And so a beetle with structural colors will remain colorful essentially forever. We found some iridescent weevils that Charles Darwin had collected in the Australian National Collection when I was working down there, and they looked exactly the same as when he would have collected them, originally. Like the structural colors do not fade. If you have an insect and you want to ask, are these colors structural or pigmentary? Do they fade after death? Then they are pigment. Do they persist after death? Then they're structural. And then back to the first part of that question was whether sexual selection could be acting upon this. The thing about beetles is that beetles don't seem to use visual cues in mating at all. They use chemical cues, they use tactile cues. Do you feel like you're the correct species to be mating with? But they don't seem to use visual cues.
Seriously?
Yeah, one of my butterflies do seem to do it. But if you look at beetles that are sexually dimorphic, when they're dimorphic, the males will have usually big weapons. They'll have horns or hooks or what have you. Sometimes they'll have huge legs with big spurs on them. Sometimes they'll have really long arms for wrestling or for grabbing onto a female, but they're usually not particularly differently colored. It's very
Give a kind of a gotcha counterexample, but I can't think of one at all.
Well, one thing we tried to do is to figure out whether beetles could see circularly polarized colors. So one of the types of multilayer reflector that you find in beetles and rutiline scarabs is circularly polarized, which means that the structure it produces can only be seen. It can be seen by people who perceive regular light, but if you were to wear those movie theater 3D glasses, the gray ones, back when we could all go to movie theaters, those are circular polarizers that go in opposite directions. And so one of those lenses will block out that color completely. And so that gives you an example of sort of an opportunity to have a visual signal that an organism that's sensitive to polarized light, to circularly polarized light, would be able to distinguish. We tried to, not we, I should say, one of my collaborators tried to figure out whether or not these scarab beetles could distinguish between polarized and unpolarized light by putting them in a little choice chamber. And the beetle didn't care at all.
Okay.
Could not have been less interested, could not tell the difference. And they really prefer to rely on chemical cues, pheromones, and tactile cues.
So my little theory that was kind of hatching in my brain as you were giving your talk was that even though they might not need high maintenance, these structural colors, they might reflect some kind of developmental symmetry, and if there's any variation among individuals, then that might reflect how kind of symmetrically organized their metamorphosis was, potentially. We've got a related question here, actually, from Sam, which is: is variation in the color heritable? Do individuals kind of inherit differences in the color?
The answer to that question is going to depend on what taxon you're looking at, I suspect. It seems to be, right now, it seems like there's not a lot of variation that we see amongst the beetles that have three-dimensional photonic crystals. Among the beetles that have multilayer reflectors, like again, the sort of bright metallic scarab beetles, we often see within a population a lot of variation in the color, because if you are a very well-fed larva, if you are very well hydrated, when you undergo pupation, the layers in your cuticle, the layers in your exoskeleton of chitin will be thicker, and you will be reflecting a longer wavelength. Conversely, if you are desiccated during pupation, if you were a small, sad little Victorian street urchin of a larva, you will have thinner cuticle, thinner layers, and you will be reflecting a shorter wavelength of light. And so, James, I don't know if you ever encountered genera like Anoplonathus or other rutiline scarabs in Australia, sort of like Christmas beetles.
Christmas beetles, yeah, they're massive white.
Yeah. And some of them, there are some species where you'll have populations that will include purple, red, green, and blue beetles. They're all forming the colors the same way, but so they just have they have variation in the thickness of those layers. So in multilayer reflecting beetles that are, have sort of that metallic iridescence, you do see quite a bit of variation in the color, but it's directly linked to development, and it doesn't appear to be heritable.
Go on then, sorry.
But that has not been formally tested, and anybody who wants to formally test it has my blessing, and I would be happy to collaborate in any way whatsoever.
There's a related question from Giovanna again. You just said that temperature, kind of desiccation, might affect the color, and so is there any kind of reverse thing? Is there any evidence that the color affects thermoregulation? And might kind of temperature dependent color be setting the beetle up for later life? That would be cool if that was adaptive.
That's a really good question, and there's actually a lovely study from one of my co-authors, Tom Schultz, from back in, I think I want to say the 70s or 80s, where he looked at tiger beetles, because tiger beetles are a fascinating example of beetle iridescence. If you look at a tiger beetle in dorsal view, it will usually look kind of a brown or white, or with a series of sort of a mottled brownish white color pattern. And when you zoom in with a microscope, you'll find that it's a pointillistic color pattern. It's actually an iridescent cuticle, an iridescent exoskeleton with this surface modification of a series of little three-dimensional pits. And those are reflecting different colors, different types of constructive interference, different wavelengths of color. And so they're creating a cryptic color using a multi-layer, an iridescent color mechanism, but not to be iridescent. Now, he looked at those guys and said, okay, I've got tiger beetles that live on mud flats and tiger beetles that live on, lake shores and what have you. Are they thermoregulating according to how much white versus black or light versus dark is on their body? His results were inconclusive, but he was also operating with not the greatest technology at the time. And so that's probably something that bears repeating. If there's anyone out there that would like to go around sticking temperature probes into furious beetles, I highly encourage it.
Fantastic. Okay. Oh yeah, that sounds amazing. Yeah, really, really good question. Finally, there's a question from Fiona. Are there any other insects that have iridescence through the same mechanism, or is this unique?
Yes, actually, there are butterflies that do it. There's 2 or 3 different families of beetles where we see three-dimensional photonic crystals, but there are also butterflies. If you see a tropical butterfly, including quite a few species from Australia, now that I think about it, with bright green or bright blue colors in the wings, that's going to be a photonic crystal. The morpho butterfly in particular has really baffled engineers throughout the ages who have been trying to recreate their specific colors. And I'm going to draw a little picture sort of explaining why. So if you look at, do I have time?
Shall I put the whiteboard on so you can?
No, that's okay. I'm just doing it on paper, and I'm just going to hold it up like caveman. So if you look at the scale of a morpho butterfly, you're going to see a series of parallel ridges. So let's
stop sharing your screen so that we can. Oh yeah. Okay.
Can you see me? So this is a wing scale of a morpho butterfly. This is about, I don't know, 250, 300 nanometers. And it's got this series of parallel ridges. So people who looked at morpho butterflies said it must be a diffraction grating. When you look at those ridges in cross section, they have this Christmas tree profile. So we said, okay, there are 2 potential diffraction gratings. There's the diffraction grating between the tips of the trees, and then there's the diffraction grating formed by the tips of the branches, for lack of a better term. And so people were trying to model morpho butterfly iridescence as the interaction of these two different perpendicular diffraction gratings. However, that's just the top of the wing scale. If you go to the bottom of the morpho butterfly wing scale, there's a photonic crystal down here, just like the beetles have.
Oh.
You've got the photonic crystal, and then in the morpho butterfly wing scales, you have these Christmas tree shaped sort of, we call them cross veins, ribbed cross veins, that are also diffracting light. And that's why morpho butterfly iridescence is so absolutely bonkers. If you were to look at I think one of the early slides in my talk has that sort of, here's an iridescent beetle, a peacock, and a morpho butterfly. And the morpho butterfly iridescence is a special case above and beyond everything else because they have 2 different forms of diffraction on top of the reflectance from these 3-dimensional photonic crystals.
Wow. So, the awesomeness of the iridescence is basically related to how many layers of diffraction there are.
Well, there's two different things going on, right? There's diffraction, and then there's this colored reflectance from the photonic crystals as well. And that's not diffraction where you're spreading the colors out. That's simply reflectance where white light comes in and only green light or only blue light comes out. Okay. And so that is very widespread amongst butterflies and some moths as well. And finally, there are some bees that also have photonic crystals.
Yes.
Seem to be only 2-dimensional. And then if anyone is really into this and wants to do a fun little literature review, we are just barely beginning to understand how spiders make their colors. If you look at those famous peacock spiders that do the little dances on YouTube, that's one of Australia's most charismatic spiders. They're just great. And they have an incredibly colorful bum that they raise up and wiggle around in a suggestive manner. And those are structural colors as well. And we are just beginning to figure out whether or not that's a photonic crystal, a diffraction grating, a multi-layer reflector, or something else. So that's really right on the cutting edge of arthropod iridescence.
That is fantastic. And so the question is, I guess, whether, well, there's no question that those mechanisms have evolved convergently, but how directly similar they are would be a really cool comparison.
Yeah, that's what we're trying to figure out. So yeah, it's really exciting. And as a taxonomist and someone who now has an entire natural history museum with a stupendously gigantic collection at their disposal, it's my preferred way to ask these questions is sort of post hoc. So take the insects that are iridescent and then work with engineers to, blast them with electrons, sand down their little scales, look inside, infer the crystal geometry. The other way to do it is bottom up, where you look at the development of it and take a transcriptomic approach and ask what genes are responsible for driving this, providing this self-assembly environment, and then creating the exact correct environment for self-assembly to progress, like making sure there's enough moisture, there's enough of the amphiphilic molecules. So there's room for genomics, there's room for transcriptomics, and then there's room for just basic good old-fashioned materials engineering.
It sounds like the grant proposal will write itself.
I know. Once we're allowed to actually go talk to other humans, I'm going to be all over the artificial and engineers. But
fantastic. Well, are there any more questions for Ainsley? Speak now or forever hold your peace. Forever hold your peace. You can ask me any questions and I can pass them on. Samantha, do you want to ask a question?
Yeah, can you hear me all right?
Yes, just fine.
Thank you. Sorry, I wanted to ask this earlier, but I'm actually having troubles finding the chat, so I just waited for a moment. It goes back to something that you were talking about earlier when you're talking about when the pupae are like soup in the chrysalis. Is there were you saying that there's no way for you to tell which cells are which when they're in there, like what they're going to turn into?
You have to pick the exact right stage of pupation. The cells that are eventually going to become the adult structures, they are there as little clusters of non-developing cells during the larval life stage. And so if you were to pick those out of the larva at a sort of a prepupal stage or an early pupation, that little imaginal disc, that little cluster of sort of pluripotent cells is going to contain the instructions for forming the entire elytron, the entire wing, the entire leg. But we just want to look at the little hairs on top of that wing, right? And so we want to wait until they've developed far enough, so about maybe halfway through pupation, so the early seta starting to develop, but before it fills out. So what you would need is someone who's an absolute wizard at TEM, at transmission electron micrographs, and someone who's very, very good at rearing these weevils so that you can get absolutely squillions of pupae that are all an exact time series of pupal development. So it could certainly be done, but it's a big technical challenge right now.
Right. So is that how you would like basically harvest this? Like you were talking about building like structures out of this chitin. Would that be like the harvesting?
I wouldn't say that would be the harvesting. It would be, you'd want to figure out chemically what's happening inside that hair. And once you could figure out what's happening, what genetic signals are happening, what is happening chemically, what are the exact concentrations of the different sort of biological materials that are present, you could then apply that to essentially, denatured chitin from other sources. Right now, most of the chitin that's being used to make chitin bioplastics is coming from shrimp, right? And so you boil down that chitin, you dissolve that back down into individual chitin building blocks, and then you should be able to reform that into a photonic crystal if you know what the chemical setup is that you need to be operating in. So that's sort of how you find out the instructions from inside the developing beetle seta during pupation, but then theoretically you'd apply that elsewhere.
Oh, very cool.
Harvest the individual hairs. So,
all right, cool. Thank you.
Yes, no problem.
Wonderful, fantastic. Well, good job, Samantha, for breaking through the audio wall. Wicked. Well, if there are no further questions for Ainsley, you can kind of get them in quickly now, or if you just think of something that was on the tip of your tongue once we've all finished, then do pass them to me, and I can pass them on to Ainsley. If not, you can follow Ainsley on Twitter, where her Twitter.
Oh no.
It's a very entertaining Twitter feed. Yeah, it just remains me to say thank you very much for an absolutely fantastic seminar. That was really, really interesting. The next one of these insect biology seminars, we've got a double whammy this week. So the next one is actually on Wednesday, so in only 2 days' time, and the speaker is me. So I'll be telling you some stuff about bee nutrition, but it looks like I've got fairly big shoes to fill with this. So I'm not sure I can follow this adequately, but I'll give it my best shot. But thank you very much, Ainsley. I don't know if you can see the chat, Ainsley, but all the students are saying thank you, thank you.
Oh, lovely. Let me look. I've opened the chat, and it's only showing my chat with James Gilbert. So yeah. All right. Well, thank you very much. It was lovely to talk to your students. They seem fantastic.
Brilliant. Well, yes, we will. I will see you on Wednesday, and Ainsley, let's catch up sometime soon. I will see you all very soon.
All right.
Thank you guys.
Cheers.
Bye, Ainsley.
Bye.